action of metaphase is to go to the middle, to be pulled to the middle. And that's exactly what happens here, except for metaphase I, we're not dealing with single chromosomes. We're dealing with the homologous chromosome pairs. We're dealing with the tetrad, but the action is still the same. The spindle fiber is still attached to the centromere, and then the centrioles kind of bring those little microtubules back in. So as they start to shorten, then or shorten or lengthen, then the chromosomes are pulled in pairs this time in the tetrads to the center of the cell. What follows metaphase? Anaphase. Same thing here. What's the action of anaphase? Away, apart. Same thing here, except for in anaphase number 1, what's being separated, we're not talking about chromatids at this point. We're not talking about parts of a chromosome. The chromosomes are still intact, but they are being pulled away from their homologous pair. So they're being separated from their homologous pair. So the tetrads separate at the centromere, and then each chromosome, so this could be chromosome number 1, this could be chromosome number 1, they're being pulled towards the opposite ends or the poles of the cell. What follows anaphase? Well, that's going to be telophase. And the action of telophase, it's the end. It is where you get 2 cells. Same thing here. So in telophase 1, cytokinesis occurs, and now we have these 2 daughter cells. And the big difference between telophase I and telophase, these daughter cells are not identical. Their chromosome number is different, and they're non-identical. They're not identical to each other, and they're not identical to the parents because of crossing over. Now here's a little section for you to complete. So dragging and dropping the information to match the picture of what just occurred in the stages of meiosis I. Now, after meiosis I, it is followed immediately by meiosis II. Because we went through a second stage of interphase, then our chromosome number would double again, and we get back up to 92. So we would only be going between this loop of 46, 92, 46, 92. But that's not going to cut it because our end game is 23 chromosomes. That's the goal. So after telophase I, we have prophase II. Again, the actions of prophase are always the action of prophase. Preparing the prep work for nuclear division. And then here's a little way that I always remember if I'm looking at a picture, if I'm looking at a picture of meiosis I, or if I'm looking at meiosis II, in meiosis I, there's just one cell going through the process. In meiosis II, there are 2 cells going through the process. So following prophase II, we go into metaphase II. And in metaphase II, what you see here, the individual chromosomes are going to line up in the middle of the cell or the equator of the cell. Now, something else happens here, and here's a big term, and we'll talk about this more in genetics, something called independent assortment. This is something else that goes on during meiosis along with that crossing over that helps to create genetic differences, genetic diversity. So you could see the chromosomes here, they're facing a particular way. Well, what if they were flipped? What if they flip over? That's going to matter because pretty soon we're going to go through telophase II, and each of these chromatids will go into an individual cell. So their positioning and how they're facing, which pole they're facing, is going to determine which egg or sperm cell they go in. So that just helps to increase that genetic diversity. So this matters. After the metaphase II, we have anaphase II. Now this looks like mitosis, except for we have 2 cells going through the process. So now we have these sister chromatids being pulled towards the opposite poles of the cell. So by these microtubules, these spindle fibers shortening, then they're being pulled towards the ends of the cell. And then to wrap it up, there's our telophase II. And in telophase 2, very different from telophase 1 in that we have 4 cells, um, and the genetic number is different. So we look at them. We have 23 chromosomes in each of these. Each of these is going to be different from the original germ cell or the original parent cell that we started out with, and each of them is going to be genetically unique from the other egg or sperm cells. And then I have a little section here for you to do the same thing you did before for meiosis 1, is to drag the descriptions of what's happening in the stage to the appropriate boxes. Then we have a little kind of compare, contrast, a little Venn diagram. Do a little drag and drop action there for mitosis versus meiosis. So if you need to go back to mitosis in order to do this, then pause me, go back to mitosis, and then come back here. Now, the last part of this is I just want to throw some terms out here for you. So the process of the cell division is meiosis. That's the process of cell division. Now, when we're talking about production of the cells themselves, what we get out of it, you might hear this process referred to as gametogenesis.